Hello students, welcome to EPG Pathshala. I am Sunil Kumar from IIT Delhi. Today we are going to talk on module Applications of Optic in Biological Instrumentation from paper Basic Biophysics. The topics which will be covered in this module are the following. We will learn about optics and optical tools that are used in biology and medicine. We will learn about some microscopy techniques and how imaging is achieved. We will discuss about fluorescence microscopy, confocal laser scanning microscopy, dark field microscopy and various other forms of microscopy techniques. We will also discuss about non-conventional microscopy techniques such as optical coherence tomography, Raman microscopy and imaging, and nonlinear optical microscopy. And briefly, we will mention how lasers are being used in various biological applications such as applications in surgery, therapy, and treatment. So at the most, mirrors, lenses, glass fibers, prisms, and gratings are the optical elements which are used very commonly in any optical instrument. We have already discussed all these optical elements in the previous module. Here we will take up a few optical instruments consisting of these elements and describe the application for which they are used in biology. We will start with optical microscopes first, explain how they work, and present a couple of examples where they have been used. We will talk about different variations of the optical microscopes that are used in biological applications. Starting our discussion with optics and optical tools. In the module before this module, we learned about light and applications of, we just introduced various applications which are possible with the light. So using optical elements such as mirrors and lenses, instruments are developed that are used for microscopy and imaging of objects, tiny objects that are difficult to visualize by naked eyes. Other most common optical components that you would find in optical instruments are optical fibers, gratings, prisms, filters, and so on. So how do you achieve imaging by a single lens? Either single lens or a combination of lenses. First, let's talk about a single lens. You might have seen handheld lenses, like as shown here in these pictures. 3 pictures where handheld lenses are used for different purposes. So what happens here when you are using a single lens? So you have basically the image created onto your retina. So there is an object on the sample plane and you place the single lens in front of it between your eye and the sample such that the image by the single lens is created on the other side of the sample and that is enlarged in size, and this image is created by your eye onto the retina. So this is the imaging which will be explained in next slide in little more detail, but this is the basic thing which happens in the case of a single lens, that it creates a magnified image of the objects. So in the imaging by a lens, let's quantify the magnification of a lens. We already understood that a single lens is used to magnify objects, but how much the magnification can be achieved by a single lens, that depends on the focal length of the lens. For example, an object of height h is placed at a distance 25 cm from your eye and you measure the height of the object to be h. You place a lens between the object and your eye such that the object is now at the focal or near the focal point of the lens. Then the object, the image of the object appears to be magnified manifolds, and the magnification is quantified as the distance of the image created by the lens divided by the focal length. That is the magnification achievable by a lens of focal length. Not necessarily a single lens is used to magnify the images magnify the size of the objects that you want to see. More often, a combination of lenses, 2 or more lenses, is used to achieve a desired magnification. For example, you have your cameras which do not use a single lens. They always use more than 2 lenses. Let's see how do we achieve magnification by a pair of lenses. As shown in this example, you have 2 lenses, 2 biconvex lenses. The first one, which is placed close to the object, is called objective lens, having a focal length f0, and another one close to your eye having a focal length fe, and the object is of certain height. Using ray optics or geometrical optics, we find that the image created by this combination of lenses is at a point I2, and the magnification, the final magnification by this pair of lenses that is appeared, that is visualized by your eye, is given by the separation between the 2 lenses, that is L, divided by the focal length of the objective lens f0 times the final distance of the object, the image from your eye, that is about 25 centimeter, divided by the focal length of the lens close to your eye, that is fe. So by playing with these 2 focal lengths, f0 and fe, and the separation between them depending on their focal lengths, one can achieve a magnification m which is controllable, which is customizable. Optical microscopes. These are actually called compound light microscope because they use more than 1 lens. Simple microscopes may have a single magnifying lens, but generally all the optical microscopes that we see around us or we have used, they have more than 1 magnifying lens, and they are called compound light microscopes. So they have either 2 or more sets of lenses. Lens that is close to your eye is called eyepiece, and the lens which is close to the object is called objective lens. Bright field light microscopes produce a dark image against brighter background, and generally all the microscopes, all the bright field microscopes provide 2-dimensional view of the object, and that is of the surface only. And in biology, commonly used to view stained cells. And the magnification of the microscope can be calculated if we know the magnification of the objective and the eyepiece sets of lenses. For example, here if you have the objective of 40x magnification and an eyepiece of 10x magnification, the total magnification of the microscope would be 400x, which means an object of size x will appear as 400 times size x. Optical microscopes, the components that are used in constructing a light microscope. In most of the microscopes, the structure is more or less the same. The optical components which are used are more or less the same. For example, as shown here in the picture, an optical microscope has a body. It has an eyepiece on the top through which you look into the microscope. Then it has a nosepiece on which 1 or more objective lenses are fixed. Then it has a sample stage on which you place your sample. Then it has adjustments for focusing the sample, to place the sample in the focal plane of the objective lenses, and there is a light source at the bottom. So this is the kind of basic configuration which you will find in any optical microscope used in labs or schools. So we understand that microscope objectives are the most important component of any microscope, which determine the final resolution or the workability or the power, resolving power of the microscope. So it becomes important to understand them a little better. So here we saw an example of a 60x objective lens from a company called Nippon. So on every objective lens you will find certain things written on it. What is the meaning of those things? For example, here Nippon is the company name or the manufacturer name. Plane means it is flat field corrected objective. Apo means it is aberration free. 60x means the maximum magnification achievable by this objective is 60 times. And the 1.40, for example, is the numerical aperture of the objective lens. Oil means the immersion medium. Most of the cases you don't put any immersion medium, and that the immersion medium which is there is simply air. But in some very special applications where you need to achieve very high resolution, there you need to a medium which has higher refractive index than air, for example water or some kind of oil, to improve the resolution. And there is another number called DIC, for example. In this case, this means dichroic property or some specialized optical property of the glass used in making the objective lens. Then you have this number infinity corrected. 0.17 is the thickness of the cover slip that can be placed below or underneath the objective to see objects. Then you have this number called WD, which is basically telling you the working distance, the maximum distance, workable distance between the same object and the microscope objective in microns. And then at the tip you have the lens, the final lens of the objective, which is closest to the object. So numerical aperture and resolution of an objective lens is explained in this slide. The numerical aperture of any objective lens is a quantity which depends on the refractive index of the immersion medium and times the angle alpha, sine of angle alpha, and alpha is the angle of the cone of the light that is accepted by the objective lens. And this numerical aperture determines the resolving power of the objective lens and the brightness of the image that is acquired. In these three pictures here, three examples are shown of objectives having different numerical apertures for the same immersion medium, meaning n is fixed. So when the numerical aperture is different, that means the acceptance angle alpha or the cone, the angle of the light cone alpha is different. In the first case, you have alpha equal to 7 degree. Any light which is outside this angle alpha doesn't reach the objective. So those objects which are away from this angle cannot be seen by the objective. So numerical aperture for this particular objective is 0.12, where n has been taken equal to 1 for air. In the second example, you have alpha equal to 20 degree. So for that particular case, you have numerical aperture equal to 0.34. And the third one, the largest one among these three, for it alpha equal to 60 degree. So larger the angle of the optical cone, larger is the Na, and that is 0.87 for this example. And then once you know the numerical aperture of the objective lens, meaning you have got a feeling, you have got an idea about the angle of the light cone accepted by the objective, that will determine your resolving power. Optical resolution is the limit below which two point-like objects placed next to one another cannot be distinguished, or they appear as one. For example, as shown in this picture, 2 point-like objects, the light coming from them is described through a function called intensity distribution function. And if we can assume it to be a Gaussian, then for the 2 points which are sitting close by, there are two Gaussian functions. So according to Rayleigh criteria, optical resolution, that is, we are talking about lateral resolution only, with a given light of lambda cannot be better than the diffraction limit. So for every wavelength of light lambda, there is a diffraction limit according to the Rayleigh's criteria. That point objects which are placed closer than the diffraction limit cannot be resolved. So resolution equals or nearly equals the wavelength of the light divided by 2 times the numerical aperture, and this resolution which we are talking about is the lateral resolution. There is another resolution which one should in principle talk about, and that is axial resolution, which is resolving objects along the axial direction, meaning along the direction of light propagation, and that is always larger than the lateral resolution which is achievable by and objective of numerical aperture NA. In this slide, we show just two examples where optical microscopes were used to image objects or microscopic objects which were difficult to see by naked eyes. In the first example, we have plant cells seen through a very high resolving power light microscope. In the second one, which might be more familiar to you, that is an optical micrograph of red blood cells. In particular case, these red blood cells were taken from a frog. They have a shape which is elliptical, and otherwise the red blood cells taken from humans, they are mostly circular in shape. So optical microscopes, depending on their resolving power or resolution, are used to image objects which are difficult to be seen by naked eyes. Let's discuss about fluorescence microscopy. What is fluorescence microscopy? So as the name suggests, the fluorescence emitted by the sample is used to image the sample. So what happens here? Light from a source excites the sample, and in most of the cases the source is a laser source, and the fluorescence from the sample at different wavelength, then the excitation wavelength is collected for imaging. So it becomes very important to separate the light which is used to excite the sample and the light coming out as a fluorescence and being used for imaging the sample, and that is done by using filters and dichroic mirrors. And generally in fluorescence microscopy, staining of the samples with specific fluorophore molecules is required. So as shown here in the image on the right hand side, this is a fluorescence microscope working in the inverted geometry, meaning you are exciting the sample from top and collecting the light which is coming in the same side of the sample. And a dichroic mirror makes sure that the light which is emitted by the sample, meaning the fluorescence light, is transmitted through it and reaches the detector. And the configuration, the experimental arrangement is such that the incident light is at 90 degrees from the direction of detection of the signal. So you will find in most of the experimental configuration arranged for fluorescence microscopy in this kind of fashion. So in fluorescence microscopy, you shine in light on the sample, and if the sample has regions which can absorb this excitation light and able to emit a light which is different from the incident light, then this sample is highly useful for fluorescence microscopy. And through electronic label picture, energy level diagram, we can understand the underlying process as shown in this picture, that you have two electronic states connected by the incident light wavelength lambda I, and then the light which is emitted by the sample is by relaxation of the molecules or the electrons from another energy level to the ground state. And because the energy separation between this final state and the ground state is smaller than the initial two states participating in the excitation process, you get a wavelength which is larger than, or energy, photons having energy smaller than the incident photons. As an example here, a picture shown where fluorescence microscopy was used to image embryonic rat hippocampal nerve cells. So here clearly the filamentous actin regions and the microtubule arrays one can distinguish, because the fluorescence properties of these two regions are different. So we are able to distinguish between the two in the fluorescence microscopy. Now we talk about fluorescence microscopy again, but in a laser scanning configuration, and that too we call it confocal laser scanning microscopy because it's not simply a laser scanning microscopy, but here By using a pinhole or some kind of aperture, we can distinguish light coming from the region where the light was focused on the sample and light coming from other regions. If we are able to collect light only from the regions where the light was focused, we can achieve imaging with very high resolution, and we can achieve imaging in 3D as well. So confocal laser scanning microscopes require a laser source for optically exciting the samples, the specimens, and the experimental configuration is quite similar to the fluorescence microscope, but the image in confocal microscopy is taken point by point scanning of the laser spot on the sample and then reconstructing the image on a computer rather than taking one full image as done in wide field fluorescence microscopy. So confocal microscopy is very useful for interior imaging because one is able to image points below the surface as well as light only from the focus regions is collected. So typical experimental setup that is used in confocal laser scanning microscopy is shown here. You have light coming from a laser source that is reflected from a dichroic mirror, then it is focused onto a pinhole, and then by using two scanning mirrors and then an objective lens, it is sent to the sample. The sample emits light, the fluorescence light in this particular example, and that fluorescence light is collected back by the objective, sent back to the dichroic mirror, and the dichroic mirror is such that it transmits only the fluorescence light, and the transmitted light is collected at a photodetector. And now by scanning this laser beam onto the sample and by moving the focal spot up and down within the sample, we collect light from different regions within the sample and then reconstruct the image of 3D volumes within the sample. As an example, images are shown here from 20 micron thick intestine sample. So in general wide field fluorescence image, you hardly see any finer details. But if you use confocal configuration, all those finer details are very clearly seen because you have achieved higher spatial resolution in the confocal configuration. Here we talk about phase contrast microscopy, which is highly useful for transparent specimens, meaning those specimens which are very difficult to see under a normal microscope because they are transparent. No light is reflected back, so you don't see anything. So here phase shifts introduced in the light passing through a transparent part of any sample is converted into brightness contrast. A phase plate is used to introduce required phase change such that the resultant change in phase is half a wavelength, and that gives you the contrast. So the principle, the mechanism behind any phase contrast microscope is presented through this cartoon here. So you have a condenser annulus just in front of light source, then you have condenser which focuses the light onto the specimen, then you have the objective, and then you have a phase plate which introduces either plus 90 or minus 90 degree phase shift. You have a gray filtering which filters the phase shifted light from the rest of the light, scattered light from the sample, and then you image the sample onto the image plane or screen, and here again you don't need to scan the whole sample. You actually get in a single shot the image of the sample. The usefulness of phase contrast microscopy, other than seeing the transparent parts of any sample, is that you not only achieve imaging of the transparent samples, you achieve more structural details also. For example, in the image shown here, you compare the images taken by a bright field microscope and a phase contrast microscope, and the differences are very clearly in terms of not only contrast but in terms of finer details as well. So the regions which are within the circles drawn on the first image, they are hardly visible in the first image, in the bright field image, but in the phase contrast microscopy you see those regions having more contrast. So this is the beauty of phase contrast microscopy, that You not only see transparent samples, but you get more structural details as well. Let's now talk about dark field microscopy. In this microscopy, a special condenser is used that creates a hollow cone of light for illuminating the sample. And the objective lens is set such that it collects only the light within the dark hollow region. So if there was no sample, there won't be any light at the image plane. But when you place the sample, there is some light which is scattered by the sample in the dark hollow region, and that light is used to image the sample. It basically creates the image of the sample at the eyepiece. As an example of dark field microscopy, here are two images for a comparison taken by a bright field microscope and a dark field microscope of a deer tick. So you see, in the bright field image, the background is bright, whereas in the dark field image, the background is dark. As we understand, in the dark field microscope, there is no light when there is no sample. But once you place the sample helps in scattering of the light, and so that the light is able to come or fill the dark hollow region that we are collecting onto the image plane. So we are able to see the objects with the help of dark field microscopy as well. So these are various forms of microscopes. You make one or few changes in a normal microscope and you can utilize the same microscope for one or different configuration for a specific application. Now we want to talk about microscopy which are different than the microscopes we talked so far. We still have the microscopic objectives used, but here the technique is entirely different than the techniques which we talked so far. We need to use an intense light source, and in optical coherence tomography, one uses scanning mirrors to scan depths in the sample and create 3D structural images of biological samples, for example. Optical coherence tomography is based on low coherence interferometry, which means that only that light is collected in a detector which has traveled a certain path length. Light beam is scanned along the axial and lateral directions to collect full 3D view of the sample. And there are many variants of optical coherence tomography technique. These can be the techniques in time domain and in Fourier domain. In the picture shown here, this is the basic configuration, basic experimental arrangement that is used in any optical coherence tomography setup. What you have is light coming from the source is divided into two parts. In one part, which is called reference arm, you have a scanning mirror, and in other arm, you have the sample placed over there. The lights coming from these two regions or these two arms is again combined at the beam splitter and sent to the photodetector. So the light which is coming from a specific depth L1 in the sample needs the scanning mirror to be placed at the proper distance from the beam splitter such that the two lights from the two arms can interfere and give you intensity at the detector. So here the graph at the bottom shows for different depths in the sample different intensity by scanning the mirrors such that the two path lengths for the two lights is matched and you get maximum intensity at the detector. For different depths, you achieve maximum intensity for different scanning lengths of the scanning mirror. So this is the basic principle which is used in every optical coherence tomography technique, that by scanning the laser beam laterally and by scanning a mirror, you can create 3D structural images of the sample under investigation. Optical coherence tomography technique was initially developed for applications in ophthalmology, and since then it has become very popular and being applied in other areas as well. In the images shown here, image A is the cross-sectional OCT image of the anterior of a human eye obtained using 1310 nanometer laser. The image B presents a 3D rendering of a 400 by 400 axial scans of the anterior eye. In the third image represents the OCT cross-sectional image of the human retina obtained at 1065 nanometer. So by looking at these pictures, you can now imagine that OCT is a technique which is used not only to image the surfaces of the samples, but to go below the surfaces as well and create 3D structural images of the samples. So highly useful for biological applications where you want to image not only the surface regions but go inside and look for crust, cavities and so on. But again, this uses a laser and some kind of arrangement which can scan the laser, and the laser which you are going to use should have a wavelength such that the wavelength can penetrate deep into the sample. That is the requirement. That is most fundamental thing one thinks of before using OCT for a specific application, for a specific sample. That the laser light which is used in the OCT should be able to penetrate much deeper into the sample. Here we talk about Raman spectroscopy, which is based on Raman scattering process, and the process is utilized in imaging as well, which has a lot of biological applications. We know that rotational and vibrational energies of molecules are very characteristic to them, meaning if you know the rotational and vibrational energies, you can guess or you can construct the structure or the bond lengths of the molecules. And if you fix your microscope for a certain wavelength, for a given wavelength, for a specific wavelength, then you can image the whole sample for that specific frequency. So what is Raman scattering? That is explained through this picture here. You shine in light on some molecule. The light has a frequency nu naught, and the molecule has a characteristic vibrational frequency given by nu1. Then the light which gets scattered from the sample can be of three types. In the first one, the light doesn't lose at all the energy. So the scattered light comes out with the same frequency, and this light is called Rayleigh scattered light. The light can lose energy, and that loss in energy is equal to the vibrational energy of the molecule. This scattered light is called anti-Stokes Raman scattered light, and the light which gets scattered at the same time can gain in energy as well, and the gain in energy is equal to the energy of the vibration of the molecule. So you have these three situations, which is pictorially explained in this picture, and through energy level diagrams, if you want to understand it, you have a ground state, and the incident light takes the molecule to some virtual state V, from where the molecules can end up and back to the ground state, or can end up in some one or the other vibrational state, and this is the case for the Stokes scattered light. And depending on this scattering, if you take a spectrum using a spectrometer, you get various lines, and these lines correspond to various vibrational or rotational states of the molecule. So each line is characteristic to the molecule. By identifying the line, you can identify the state of vibration or a state of rotation of the molecule. And now the idea in Raman imaging is that you pick any one of these lines in the spectrum and fix your microscope in such a manner that you image the sample only for this frequency, and that is what gives you Raman imaging. So Raman imaging is highly useful in biology for detection of tumors or cancerous cells. Why it is so popular in biology is because in regions where there is tumor or the cells are cancerous, Raman frequencies which you see in the Raman spectrum, they are shifted slightly. Now, as you said before, if you are scanning, if you are imaging the sample for a particular frequency, and because of cancerous cells the frequency has slightly shifted, you will not see anything in that region where there are cancerous cells. As an example, the images shown here are taken on a mouse brain. The image on the left hand side is taken by a bright field image, and the image on the right hand side is taken by a Raman microscope where the imaging was done at a frequency of 2900 or around that centimeter inverse. So what you see is, you see a region where there is no signal at all at that frequency. So why it is so? It is so because in that particular region the frequency of vibration of those molecules or those cells which are all around here in this particular sample has changed, and this could happen because of cancer or development of a tumor. So this is the powerfulness of a Raman imaging setup, that this technique can be used for detection of cancerous cells. Now let's talk about little more than the microscopy which we talked so far, that is non-linear optical microscopy. So far we were just discussing the linear response of the material that we wanted to image with respect to the light that was incident on it. So higher the intensity, higher was the brightness that you could see. But here we have the interaction of light and material slightly different than linear. We call it non-linear because the response is simply not proportional to the intensity of the incident light, to the amount of the field of the incident light. Rather, it is dependent on either the intensity or square of the intensity or higher powers of the intensity of the incident light. And because more number of photons, so every intensity or every light that is incident on it in terms of photons, if in one interaction there are two or more photons involved, either in the scattering process or in the absorption process, then that process becomes nonlinear process. And some of these processes have been found highly useful for microscopy and imaging applications, because such processes are dependent on the intensity and higher orders of intensity, meaning the electric field associated with the light that is incident on the sample, from the fact that intensity is square of the electric field associated with the light, we need to have a light which is strong enough so that the higher orders responses from the sample can be comparable with the linear response of the sample, and this nonlinear response is then used for imaging. So you need to have light coming from some kind of laser source in the form of pulses, because when you have pulses of lights, you have the electric field associated with it comparably quite high. So nonlinear interaction between light and matter is used as the contrast mechanism, and technical details of the experimental setups are otherwise same as those discussed before in laser scanning microscopy techniques. And then which nonlinear interaction is used for microscopy, that is decided by the spectral selection of the light that is emitted or scattered by the sample. So as we said, that nonlinear interaction is used for imaging and the intensity as the contrast mechanism. So two or more photon transitions are involved in the laser excitation process, and depending on the photon that is detected, the techniques can be second harmonic generation as shown here, and the two photon which combine in the sample and give you a single photon shown in blue is the second order photon. Or the technique can be based on third harmonic generation, meaning three incident photons combine and give you a single photon of 3 times higher frequency. Or the nonlinear process involved can be two photon excited fluorescence. So as an example of nonlinear optical imaging of human skin taken in the second harmonic generation and the two photon excited fluorescence modes. So in the second harmonic image, you just see the collagen bundles, whereas in the two photon excited fluorescence image, you see only the elastic fibers present in the skin. And together, the SHG and the two photon excited fluorescence images, the overlay of these two is shown in figure C, and the difference, the contrast is amazing, that every nonlinear process is able to detect a different thing. and that is the power of nonlinear optical microscopes. That every nonlinear process is used to detect a different. Now let's finally point out some applications of lasers in surgery and therapy, because laser is the device, or that is the invention which gave you a lot of energy, lot of intensity, meaning lasers are the intense source of light, and therefore lasers have found very common use in imaging and microscopy applications. In industry as well, high power lasers are being used for marking, cutting, welding and fusor applications. In biology, not only microscopy and imaging is achieved by lasers, but lasers are being used for surgery and therapy applications as well. Some of the examples in biology where lasers are being used extensively are LASIK for eye surgery and LASIK and so on. Let's quickly review what we learned so far in this module through some questions. The first question we would like to ask is the following. Out of the following choices for the sources, which can provide light for the deepest penetration into the biological sample? Will it be an ultraviolet source, or a visible source, an X-ray source, or a far infrared source? Second question would be, which of the following objective lens will provide the highest resolution? We are talking about here an objective with numerical aperture 0.1 or 0.5 or 0.05. Third question that we would like to ask is, all the microscopy techniques that we discussed in this module are based on either geometrical optics, wave optics or light photons. So out of these three options, which do you think most suitable to explain all the microscopy techniques that we discussed in this module? So these were the questions to review what we learned in the module. So students, let us summarize what we have learned in this module. We learned that optical microscope is an instrument that is used to see objects difficult to observe with naked eyes. And these optical microscopes can be found in various forms. It all depends on the components which are used to construct the optical microscope and the layout. So depending on the requirements, one can modify the existing optical microscope to perform either bright field microscopy or dark field microscopy or fluorescence microscopy or phase contrast microscopy. Laser scanning confocal microscope is a special form of fluorescence microscope, where a laser is used to optically excite the sample under observation, and a pinhole placed in the excitation path ensures that light coming from only the focal point region within the sample reaches the camera. Raman microscope is similar to fluorescence microscope in construction, but it has the advantage that one can obtain molecular vibration specific images. Therefore, in detection of cancer or other diseases, Raman microscopy is highly useful. Optical coherence tomography is one of the most popular techniques in ophthalmology, where one can create 3D scanning images at very high speeds. Using high power lasers, there are nonlinear optical microscopes which are designed to bring different types of contrast mechanisms in the sample imaging. In this, pulse lasers are used, which are in demand these days because they are highly useful in surgery, therapy and treatment.